Beneath the forest floor, where the light of the sun never reaches, lies a vast and silent intelligence that has been breathing for 1 billion years, waiting for us to finally fall silent and listen. This is the mycelium, the hidden body of the fungus, a network of thread-like filaments called hyphae that weave through soil and wood and leaf litter, branching and merging and extending in every direction, forming connections that span acres, sometimes miles, creating an underground web of communication and exchange that links tree to tree, plant to plant, organism to organism in a living internet older than any forest, older than any root, older than the first plant ever dared to leave the water and colonize the land. The mycelium is not a single organism but a colony, a collective intelligence distributed across billions of microscopic threads, each one thinner than a human hair, each one growing at the tip, secreting enzymes, dissolving organic matter, absorbing nutrients, and sending chemical signals through the network. The hyphae branch and fuse, creating a mesh so dense that a single teaspoon of healthy forest soil can contain miles of fungal filaments, invisible to the naked eye, yet essential to the life of the forest, to the cycling of nutrients, to the health of the trees that tower above. Mycelium does not think as we think. It has no brain, no central nervous system, no neurons firing in coordinated patterns. Yet it responds to its environment with a sophistication that suggests awareness, intention, memory. When a hyphae encounters a food source, it grows toward it, extending its tip, secreting enzymes to break down the material, absorbing the nutrients. When it encounters a toxin or a competitor, it withdraws, redirects its growth, or releases defensive compounds. When the network is damaged, it reroutes, finding new paths, maintaining the flow of nutrients and information. The mycelium is patient. It can wait for years, decades, centuries, lying dormant in the soil, its metabolism slowed to a crawl, conserving energy, waiting for the right conditions, moisture, warmth, the presence of a host or a food source. When the conditions are met, it awakens, growing rapidly, extending its network, forming fruiting bodies, the mushrooms we see above ground, the reproductive structures that release spores into the air, continuing the cycle. But the mushroom is only the fruit. The mycelium is the tree. The mycelium explores. When a hyphae extends into unknown territory, it does not grow in a straight line but branches, sending out multiple tips in different directions, testing the environment, searching for resources. If one branch encounters favorable conditions, a rich source of nutrients, optimal moisture, the right temperature, it grows more vigorously, allocating more resources to that direction. If another branch encounters poor conditions or competition, it slows, withdraws, or dies back, conserving energy for more promising paths. This is foraging behavior, decision-making distributed across thousands of hyphal tips, each one acting semi-autonomously yet contributing to the collective strategy of the network. The mycelium learns from its explorations. Studies have shown that fungal networks can optimize their growth patterns over time, becoming more efficient at locating and exploiting resources. When presented with food sources arranged in a pattern, the mycelium will initially grow in all directions, exploring randomly. But over successive generations, the network refines its architecture, concentrating growth along the most efficient pathways, minimizing redundancy, creating a structure that resembles engineered transportation networks, subway systems, highway grids, neural pathways. The mycelium arrives at these solutions not through conscious design, but through trial and error, through the accumulation of small local decisions that aggregate into global patterns of remarkable efficiency. The mycelium communicates. Recent research has revealed that fungal networks can transmit signals between plants, warning of insect attacks, sharing nutrients, even influencing the behavior of neighboring trees. When a tree is attacked by aphids, it releases chemical signals into the air, warning nearby trees to activate their defenses. But the signal also travels underground, through the mycelial network, reaching trees that are not connected by air, trees that are shaded, trees that would not otherwise receive the warning. The mycelium acts as a conduit, a living telegraph system carrying messages across the forest floor. The signals are chemical, electrical, and possibly more. Hyphae generate electrical impulses as nutrients and water flow through them, creating patterns of activity that resemble the neural activity of animal brains. Some researchers have measured these impulses and found that they vary in frequency and amplitude depending on environmental conditions, suggesting that the mycelium is processing information, integrating signals from multiple sources, and responding in coordinated ways. The electrical signals travel through the network at speeds of up to 1 millimeter per second, slow by the standards of animal nervous systems, but fast enough to coordinate responses across the network, to synchronize fruiting, to redirect growth in response to threats or opportunities. Whether this constitutes intelligence in any meaningful sense is debated, but the complexity of the network, the sophistication of its responses, and the scale of its operations suggest that the mycelium is far more than a passive absorber of nutrients. It is an active agent, shaping its environment, making choices, solving problems. The intelligence is distributed, emergent, arising from the interactions of countless simple components, each following local rules, yet producing global behaviors that appear purposeful, adaptive, even strategic. The largest known organism on Earth is a fungus. Armillaria ostoyae, the honey mushroom, forms a mycelial network in the Blue Mountains of Oregon that covers over 2,000 acres and is estimated to be between 2,000 and 8,000 years old. The network is a single genetic individual, a clone that has been growing and spreading for millennia, consuming dead wood, infecting living trees, and fruiting in the autumn, producing clusters of golden mushrooms that emerge from the soil and the bases of trees. The organism weighs hundreds of tons, perhaps thousands, and it is still growing, still expanding, still alive. This is not an anomaly. Many fungi form extensive networks, and some are even larger, though less well documented. The mycelium does not respect boundaries. It flows through the soil like water, following gradients of nutrients and moisture, connecting disparate parts of the ecosystem, creating a web of interdependence that is invisible to those who walk above. The mycelium is ancient. Fungi are among the oldest eukaryotic organisms, with fossil evidence dating back over 1 billion years. They were among the first organisms to colonize land, forming symbiotic relationships with early plants, helping them extract nutrients from barren rock, facilitating the transition from aquatic to terrestrial life. Without fungi, there would be no forests, no grasslands, no complex terrestrial ecosystems. The mycelium is the foundation upon which the green world is built. The structure of the mycelium is fractal, self-similar at different scales, branching in patterns that maximize surface area and minimize energy expenditure. The network is efficient, resilient, and adaptive. If a section of the mycelium is destroyed, the network reroutes, finding alternative paths, maintaining connectivity. If resources are abundant in one area, the mycelium grows more densely there, concentrating its efforts. If resources are scarce, it extends farther, searching, exploring, colonizing new territory. The mycelium is also a recycler, a decomposer, an agent of transformation. It breaks down dead organic matter, fallen leaves, dead wood, animal carcasses, releasing nutrients back into the soil, making them available to plants, to bacteria, to other fungi. Without decomposers, the world would be buried in dead matter, nutrients locked away, unavailable to the living. The mycelium is the engine of decay, the force that turns death into life, that closes the loop, that ensures the cycle continues. But the mycelium is not merely a passive decomposer, it is selective, strategic, even competitive. Different species of fungi specialize in breaking down different materials. Some digest cellulose, others lignin, others chitin or keratin. Some fungi are generalists, able to consume a wide range of organic matter. Others are specialists, adapted to specific substrates, specific environments, specific hosts. The mycelium competes with bacteria, with other fungi, with insects and nematodes, all vying for the same resources, all engaged in a silent, slow-motion struggle for survival. The mycelium also forms alliances. Mycorrhizal fungi, which associate with the roots of plants, are among the most important symbiotic organisms on Earth. They extend the reach of the plant's root system, increasing its ability to absorb water and nutrients, particularly phosphorus and nitrogen, which are often limiting in natural soils. In exchange, the plant provides the fungus with sugars, the products of photosynthesis, which the fungus cannot produce on its own. This exchange is ancient, dating back over 400 million years, and it is essential to the survival of most terrestrial plants. Over 90% of plant species form mycorrhizal associations, and many cannot survive without them. The mycorrhizal network is sometimes called the Wood Wide Web, a term that captures the interconnectedness of the forest, the way trees and fungi and other plants are linked in a vast underground network of exchange and communication. The network is not egalitarian. Some trees are hubs, highly connected, receiving and distributing resources to many other trees. Others are peripheral, less connected, more dependent on the generosity of the network. Older trees, with more extensive root systems and larger mycorrhizal networks, often support younger trees, sharing carbon and nutrients, helping them survive in the shade, in poor soil, in times of stress. This sharing is not altruism, at least not in the human sense. The trees and fungi are engaged in a mutualistic relationship, each benefiting from the exchange. But the network also creates a form of interdependence, a collective resilience, where the health of one organism is linked to the health of others, where the forest functions as a superorganism greater than the sum of its parts. The mycelium senses. It detects gradients of nutrients, of moisture, of oxygen. It responds to touch, to chemical signals, to the presence of other organisms. Some fungi can even sense light, though they do not photosynthesize, using light as a cue for fruiting, for orienting their growth, for timing their reproductive cycles. The mycelium is embedded in its environment, constantly sampling, constantly adjusting, constantly responding to the conditions around it. The mycelium remembers. Studies have shown that fungi can exhibit a form of memory, altering their growth patterns based on past experiences. A mycelium that has encountered a toxin in one location will avoid that location in the future, even if the toxin is no longer present. A mycelium that has found a rich food source will return to that location, even if the food has been consumed, searching for more. This is not conscious memory, not the recall of specific events, but a form of learned behavior, encoded in the structure and chemistry of the network, a memory written in the branching patterns of hyphae, in the distribution of resources, in the pathways that have been reinforced by repeated use. The mycelium operates on timescales that dwarf human perception. A single hypha can extend several micrometers per minute, imperceptible to the naked eye. Yet over days and weeks, the network can expand across meters, colonizing new territory, connecting distant resources. The fruiting bodies, the mushrooms, can emerge overnight, pushing through soil and leaf litter with surprising force, expanding from pinheads to full-sized caps in a matter of hours. But this rapid growth is the culmination of weeks or months of preparation, of nutrient accumulation, of environmental sensing, of waiting for the precise combination of temperature, moisture, and light that signals the right moment to fruit. The mycelium engages in chemical warfare. Fungi produce an astonishing array of secondary metabolites, compounds that are not essential for basic metabolism, but serve defensive, competitive, or communicative functions. Antibiotics like penicillin, discovered in the fungus Penicillium, are weapons that fungi use to suppress bacterial competitors in the soil. Other fungi produce toxins that deter predators, insects, nematodes, slugs, that might otherwise consume the mycelium or the fruiting bodies. Some fungi produce allelopathic compounds that inhibit the growth of competing fungi, creating zones of exclusion around their networks. The soil is a battlefield, and the mycelium is armed with chemistry, deploying molecular weapons in a slow, silent war for resources and territory. Yet the mycelium is also cooperative, forming alliances that transcend species boundaries. Lichens are composite organisms, symbiotic partnerships between fungi and photosynthetic algae, or cyanobacteria. The fungus provides structure and protection, absorbing water and minerals from the substrate. The photosynthetic partner provides sugars, the products of photosynthesis. Together, they can colonize environments that neither could survive alone: bare rock, desert soil, Arctic tundra, the bark of trees. Lichens are among the oldest living organisms, with some individuals estimated to be thousands of years old, growing millimeters per year, patient beyond measure. The mycelium is everywhere, in forests, in grasslands, in deserts, in tundra, in the soil of gardens and farms, in the wood of houses and fences, in the air, in the water, in the bodies of plants and animals. Fungi are ubiquitous, diverse, essential. They are the hidden majority, the unseen workers, the silent architects of ecosystems. They do not demand attention, do not announce their presence, do not compete for the light. They work in darkness, in silence, in the spaces between, connecting, transforming, sustaining. And beneath the forest floor, where the light of the sun never reaches, the mycelium continues its work, growing, branching, communicating, recycling, supporting, a vast and silent intelligence that has been breathing for 1 billion years, waiting for us to finally fall silent and listen. The network hums with life, with chemistry, with the slow, patient exchange of nutrients and signals, a conversation that has been ongoing since before the first tree stood upright, since before the first leaf unfurled, since before the land was green. The mycelium is the foundation, the substrate, the living matrix upon which the visible world rests, and it is still here, still growing, still connecting, still waiting. Death is not an ending in the forest. It is a transformation, a slow unraveling of complex structures back into their constituent elements, a process that would take centuries if left to physical forces alone, but is accelerated to years, months, sometimes weeks by the patient work of fungi, the great recyclers, the decomposers that stand between the living and the dead, breaking down what was once alive, releasing nutrients locked in wood and leaf and flesh, returning them to the soil where they can be taken up by roots, incorporated into new growth, woven into the fabric of new life. Without fungi, the world would be buried under its own dead, forests choked with fallen trees, nutrients trapped in inert matter, the cycle of life grinding to a halt. Fungi are the only organisms capable of fully decomposing wood. Cellulose, the primary structural component of plant cell walls, can be broken down by many organisms, bacteria, insects, some animals. But lignin, the complex polymer that gives wood its strength and rigidity, is nearly indestructible, resistant to most forms of decay, impervious to water, to acids, to the enzymes that break down other organic molecules. Only fungi possess the enzymatic machinery to dismantle lignin, to cleave its aromatic rings, to oxidize its carbon-carbon bonds, to reduce it to simpler compounds that can be metabolized or released back into the environment. The process is slow, methodical, relentless. A fallen tree lying on the forest floor becomes a nursery for fungal life. Spores land on the bark, germinate, send hyphae into the wood, following cracks and fissures, penetrating the cell walls, secreting enzymes that dissolve the lignin and cellulose, absorbing the sugars and nutrients released. The wood softens, darkens, becomes spongy, crumbling under pressure. Fruiting bodies emerge, brackets, shelves, clusters of mushrooms, each one releasing millions of spores that drift on the wind, seeking new wood, new opportunities, continuing the cycle. The fungi that decompose wood are called saprotrophs, organisms that feed on dead organic matter. They are specialists adapted to specific substrates, specific stages of decay. Some fungi colonize freshly fallen wood, attacking the sapwood, the outer layers rich in sugars and starches. Others wait, colonizing wood that has already been partially decomposed, feeding on the lignin-rich heartwood that earlier colonizers could not fully digest. Still others arrive late, feeding on the remnants, the humus, the final stages of decay when the wood has been reduced to a crumbly soil-like substance that blends into the forest floor. This succession of fungi, each species replacing the last, is a form of ecological relay, a passing of the baton from one decomposer to the next, each one contributing to the gradual transformation of wood into soil. The stages of decay are distinct, measurable, predictable. Fresh wood is hard, intact, its cells still holding their shape. As fungi colonize it, the wood enters the first stage of decay. Slight softening, some discoloration, the bark beginning to loosen. In the second stage, the sapwood becomes spongy, the wood loses structural integrity, and the first conchs and brackets appear, shelf-like fruiting bodies that protrude from the trunk like shelves from a wall. By the third stage, the wood is soft enough to crumble in the hand, riddled with tunnels and galleries carved by insects that follow the fungi, feeding on the partially digested wood, accelerating the decay. The fourth stage sees the wood reduced to fragments, to chunks that break apart easily, that blend into the leaf litter, that are colonized by mosses and ferns and seedlings that root in the decaying mass. In the final stage, the wood is gone, transformed into humus, into soil, into the dark, rich substrate that feeds the next generation of trees. The process can take decades for a large tree, centuries for the most decay-resistant species. Redwood, cedar, oak, but eventually the wood disappears, its atoms dispersed, its energy dissipated, its structure dissolved, leaving behind only a faint depression in the soil, a nurse log that has nurtured seedlings, mosses, ferns, and fungi, giving life even as it decayed. The fallen tree becomes a cradle for new life, a horizontal garden suspended above the forest floor, a monument to the power of decomposition to transform death into fertility. Fungi do not only decompose wood, they break down leaves, needles, twigs, roots, flowers, fruits, seeds, the endless rain of organic matter that falls to the forest floor each autumn. Leaf litter is a buffet for fungi, each species specializing in different components. Some digest the cellulose, others the proteins, others the waxes and resins that coat the leaf surface. The litter is transformed layer by layer into humus, a dark, nutrient-rich substance that improves soil structure, retains moisture, and provides a reservoir of nutrients for plants. Fungi also decompose animal matter, carcasses, bones, feathers, fur, chitin. Carrion fungi colonize dead animals, breaking down proteins and fats, releasing ammonia and other nitrogen compounds into the soil. Some fungi specialize in keratin, the protein that makes up hair, nails, hooves, and horns, materials that are resistant to most forms of decay. Others digest chitin, the structural polymer of insect exoskeletons and fungal cell walls, recycling it back into the ecosystem. The decomposition process releases nutrients in forms that plants can absorb: nitrogen as nitrate or ammonium, phosphorus as phosphate, sulfur as sulfate, along with trace elements like iron, magnesium, calcium, and potassium. These nutrients are taken up by plant roots, often with the assistance of mycorrhizal fungi, and incorporated into new tissues, new leaves, new wood. When the plant dies, the nutrients are released again, and the cycle continues, an endless loop of growth and decay, of synthesis and decomposition, mediated by fungi. The enzymes that fungi use to decompose organic matter are among the most powerful catalysts in nature: lignin peroxidases, manganese peroxidases, laccases. These are oxidative enzymes that generate free radicals, highly reactive molecules that attack the chemical bonds in lignin, breaking them apart. The process is non-specific, aggressive, capable of degrading not only lignin but also a wide range of other complex organic molecules, including pollutants, pesticides, and industrial chemicals. This has made fungi valuable in bioremediation, the use of living organisms to clean up contaminated environments. Fungi have been used to degrade oil spills, breaking down hydrocarbons into less toxic compounds. They have been used to remove heavy metals from soil, accumulating the metals in their tissues, which can then be harvested and disposed of safely. They have been used to degrade plastics, though the process is slow and not yet practical on a large scale. The same enzymes that evolved to break down lignin, one of the most recalcitrant natural polymers, can also attack synthetic polymers, given time and the right conditions. The decomposition of organic matter by fungi is not just a chemical process, but an ecological one, creating habitats, supporting food webs, shaping the structure of ecosystems. A rotting log is a microhabitat, home to insects, spiders, salamanders, small mammals, each one dependent on the fungi that are breaking down the wood, creating cavities, softening the substrate, providing food and shelter. The fruiting bodies of fungi are food for slugs, snails, beetles, flies, and mammals, deer, squirrels, bears, that disperse the spores, spreading the fungi to new locations. Fungi also play a role in the carbon cycle, one of the most important biogeochemical cycles on Earth. When plants photosynthesize, they capture carbon dioxide from the atmosphere and convert it into organic compounds, sugars, cellulose, lignin. When plants die, the carbon is stored in their tissues, and if the tissues are not decomposed, the carbon remains locked away, sequestered from the atmosphere. Fungi release this carbon, breaking down the organic matter and respiring carbon dioxide back into the atmosphere, completing the cycle. But not all carbon is released. Some of it is converted into fungal biomass, into mycelium and fruiting bodies, which are themselves eventually decomposed by other fungi, other bacteria, other organisms. Some of it is incorporated into humus, a stable form of organic matter that can persist in the soil for decades, centuries, even millennia, slowly releasing nutrients and carbon over time. The balance between carbon sequestration and carbon release is influenced by many factors: temperature, moisture, the composition of the organic matter, the diversity and activity of the decomposer community. In cold climates, decomposition is slow and organic matter accumulates, forming thick layers of peat, of leaf litter, of partially decomposed wood. The fungi work slowly in the cold, their enzymes less active, their metabolism slowed, their growth rates reduced. A fallen tree in a boreal forest might take a century to fully decompose, its wood preserved by the cold, by the acidity of the soil, by the scarcity of the specialized fungi that can break down lignin in such harsh conditions. The forest floor becomes a graveyard of logs, each one in a different stage of decay, each one supporting a different community of fungi, insects, and other organisms. In tropical climates, decomposition is rapid, and organic matter is recycled quickly, leaving little accumulation on the forest floor. The heat and humidity accelerate enzymatic activity, and the diversity of decomposer fungi is staggering. Hundreds of species can be found on a single fallen tree, each one occupying a specific niche, each one contributing to the rapid breakdown of the wood. A tree that falls in a tropical rainforest might be reduced to soil in a matter of years, its nutrients released and taken up by the surrounding vegetation almost as quickly as they are freed from the decaying wood. The rate of decomposition affects the fertility of the soil, the productivity of the ecosystem, and the amount of carbon stored in the soil versus the atmosphere. In ecosystems where decomposition is slow, carbon accumulates in the soil, sequestered from the atmosphere for decades or centuries. In ecosystems where decomposition is rapid, carbon is quickly returned to the atmosphere as carbon dioxide, completing the cycle but contributing to the greenhouse effect. The balance is delicate, influenced by temperature, moisture, the chemistry of the organic matter, and the diversity and activity of the decomposer community. The diversity of decomposer fungi is itself a form of resilience. Different species respond to different environmental conditions, different substrates, different stages of decay. When conditions change, when a drought occurs, when temperatures fluctuate, when new types of organic matter become available, the fungal community shifts, with some species becoming more abundant and others declining. This flexibility ensures that decomposition continues, that nutrients continue to be recycled, that the ecosystem remains functional even in the face of environmental variability. Fungi are also involved in the nitrogen cycle, converting organic nitrogen, proteins, amino acids, nucleic acids into inorganic forms that plants can use. Some fungi form symbiotic relationships with nitrogen-fixing bacteria, facilitating the conversion of atmospheric nitrogen into ammonia. Others decompose nitrogen-rich materials, releasing ammonia and nitrate into the soil. The availability of nitrogen is often a limiting factor for plant growth, and fungi play a crucial role in making nitrogen available, recycling it from dead matter, transferring it through mycorrhizal networks, supporting the productivity of ecosystems. The work of decomposition is invisible, unglamorous, essential. It happens in darkness, in dampness, in the hidden spaces beneath the forest floor, in the crevices of rotting logs, in the layers of leaf litter. It is slow, patient, thorough. A mushroom that appears overnight as the culmination of weeks of unseen labor, of hyphae spreading through the substrate, of enzymes dissolving complex molecules, of nutrients being absorbed and transported, of energy being accumulated and stored, waiting for the right moment to fruit, to reproduce, to send spores into the world. And when the fruiting body decays, when the mushroom collapses and dissolves back into the soil, it too is decomposed, its nutrients recycled, its carbon released or stored, its atoms redistributed, continuing the cycle. Nothing is wasted. Nothing is lost. Everything is transformed, broken down, rebuilt, reused, in an endless circulation of matter and energy that has been ongoing for billions of years, since the first organisms began to die and the first decomposers evolved to consume them. The fungi are the custodians of this cycle, the agents of transformation, the bridge between death and life. They do not create nutrients, they release them. They do not generate energy, they transfer it. They do not build ecosystems, they sustain them by ensuring that the dead do not accumulate, that nutrients do not become locked away, that the cycle continues, that life can persist, and in the quiet of the forest, where a fallen tree lies slowly dissolving into the earth, where mushrooms emerge from the bark in silent profusion, where the scent of decay mingles with the scent of growth, the fungi continue their work, patient, persistent, essential, turning death into life, one molecule at a time. The great recyclers do not rest. They are always present, always active, always waiting for the next leaf to fall, the next tree to topple, the next opportunity to transform what was into what will be. They are the unseen workers, the silent partners, the essential agents of renewal, ensuring that the forest, the grassland, the soil, the world remains fertile, productive, alive. And beneath it all, the mycelium spreads, connecting the living and the dead, the growing and the decaying, the past and the future, in a web of transformation that spans the forest floor, that reaches into every corner of the ecosystem, that has been operating since before there were forests, since before there were trees, since the first fungi learned to break down the first dead cells and return their substance to the world. 400 million years ago, long before the first tree stood upright, long before the first flower bloomed, long before the first insect took flight. The land was dominated by organisms that towered over the primitive plants that clung to the edges of rivers and lakes, massive columnar structures that rose 8 meters into the air, trunks a meter wide, tapering slightly toward the top, their surfaces textured with a pattern of interwoven filaments, their interiors composed of densely packed tubes arranged in concentric rings. These were prototaxites, the giants of the Silurian and Devonian periods, and for over a century after their fossilized remains were first discovered, scientists debated what they were: trees, algae, lichens, or something else. Entirely, they were fungi, the largest organisms on land at a time when nothing else grew taller than a meter. The realization came slowly, reluctantly, through the accumulation of evidence that could not be explained by any other hypothesis: the structure of the fossils, the arrangement of tubes, the absence of leaves, roots, or branches. The pattern of growth did not match any known plant. The chemical composition of the preserved tissues, analyzed through isotopic studies, revealed ratios of carbon isotopes consistent with a fungal metabolism, not photosynthesis. The anatomy, examined under microscopes, showed hyphae, the thread-like filaments characteristic of fungi, woven together in a dense, three-dimensional matrix. Prototaxites was a fungus, but unlike any fungus alive today. It did not fruit and release spores from a small ephemeral mushroom. It was the fruiting body, a massive perennial structure that stood for years, perhaps decades, slowly growing, slowly releasing spores from its surface or from pores near the top. Or perhaps it was not a fruiting body at all, but a vegetative structure, a vertical extension of the mycelium, a tower built to elevate spores above the ground, to catch the wind, to disperse them across the barren landscape. The world in which prototaxites lived was alien, unrecognizable. The land was largely barren, covered with mats of primitive plants, mosses, liverworts, early vascular plants like Cooksonia, small and simple, lacking true leaves or roots, spreading horizontally across the ground. There were no forests, no trees, no flowers, no grasses. The atmosphere contained less oxygen than today, and the ozone layer was thinner, allowing more ultraviolet radiation to reach the surface. The climate was warm, the continents were arranged differently, and the seas teemed with life, trilobites, brachiopods, early fish. But the land was still being colonized, still transitioning from a lifeless expanse of rock to a living ecosystem. In this world, prototaxites stood like monuments, like pillars, like the remnants of some ancient architecture. They grew in clusters in groves, their trunks rising from the ground in silent profusion, their surfaces weathered by wind and rain, their interiors slowly accumulating mass, layer by layer, year by year. They were the tallest structures on land, visible from a distance, landmarks in a landscape otherwise flat and featureless. Imagine walking through this ancient world, through a landscape where the sky stretches unbroken to the horizon, where the ground is covered with low mats of green and brown, where the air smells of damp earth and algae, where the only sounds are the wind and the distant crash of waves. And rising from the ground, scattered across the plain, are the trunks of prototaxites, massive columns of woven fungal tissue, their surfaces rough and fibrous, their colours muted, browns, greys, perhaps tinged with green from algae or lichens growing on their surfaces. Some are young, barely a metre tall, still growing. Others are ancient, 8 metres high, their tops weathered and broken, their bases thick and gnarled. The fossils tell fragments of this story. Cross-sections reveal the internal structure, concentric rings of tissue, like tree rings but made of fungal hyphae, densely packed interwoven, creating a matrix strong enough to support the weight of the column. The outer surface shows a pattern of interlacing filaments, a texture unlike any modern organism. Some fossils preserve the base of the trunk, showing how it spread across the ground, anchoring the structure, drawing nutrients from the soil or from the organic matter that accumulated around it. What did they feed on? The question is difficult to answer because the ecosystem was so different from anything that exists today. There were no trees to decompose, no thick layers of leaf litter, no abundant sources of dead organic matter. The most likely hypothesis is that prototaxites was saprotrophic, feeding on the remains of the primitive plants that grew around it, on the mats of algae and cyanobacteria that covered wet soils, on the organic matter that accumulated in shallow pools and along the edges of rivers. Or perhaps it was symbiotic, forming associations with photosynthetic organisms, algae, cyanobacteria, early plants, exchanging nutrients for sugars, much like modern mycorrhizal fungi. Another hypothesis suggests that prototaxites was a lichen, a composite organism formed by the symbiosis of a fungus and a photosynthetic partner. The structure of the fossils, with their dense interwoven filaments, is consistent with the anatomy of some modern lichens, and the size could be explained by the abundance of moisture and the lack of competition from other organisms. But the isotopic evidence argues against this, suggesting that prototaxites obtained its carbon from decomposing organic matter, not from photosynthesis. The truth may never be fully known. Prototaxites left no living descendants, no modern analogues that can be studied to infer its biology. It existed for over 50 million years, from the mid-Silurian to the late Devonian, and then it vanished from the fossil record, disappearing as the first forests began to grow, as vascular plants diversified and spread, as the land became greener, more complex, more competitive. The giants were replaced by trees, and the fungi that had once towered over the landscape retreated into the soil, into the wood, into the shadows, becoming decomposers, symbionts, parasites, but never again achieving the stature they had once held. The disappearance of prototaxites coincides with the rise of the first forests, the spread of plants with true roots, stems and leaves, the development of lignin, the complex polymer that gives wood its strength and allows trees to grow tall. The forests changed the land, creating shade, altering the climate, increasing the amount of organic matter that accumulated on the ground. The fungi adapted, evolving new strategies, new enzymes, new relationships. They became the decomposers of wood, the partners of trees, the recyclers of nutrients. They became smaller, more diverse, more specialized, occupying niches that had not existed when prototaxites stood alone. But the legacy of prototaxites remains, a reminder that fungi were once the dominant organisms on land, that they shaped the early terrestrial ecosystems, that they were the pioneers, the colonizers, the the giants. The fossils are rare, found in a few locations around the world: Scotland, Canada, Saudi Arabia, Australia, preserved in sedimentary rocks that formed in ancient river deltas, in coastal plains, in environments where the conditions were just right for fossilization. The preservation is often exquisite, with details of the internal structure visible under microscopes, revealing the arrangement of hyphae, the patterns of growth, the architecture of an organism unlike anything alive today. The fossils are often fragmentary, cross-sections of trunks, impressions of surfaces, pieces of a puzzle that scientists have been trying to assemble for over a century. Some specimens are complete enough to show the full height of the organism, the taper from base to top, the texture of the surface. Others are just fragments, chunks of fossilized tissue that hint at the size and structure, but leave many questions unanswered. The size of prototaxites is staggering when considered in context. 8 meters tall, in a world where the tallest plants were less than a meter. It would be like a human standing among organisms the size of house cats, a giant among pygmies, a structure so large that it defied the scale of everything around it. The trunks were massive, solid, built from densely packed hyphae that gave them structural integrity, that allowed them to stand upright, to resist wind and weather, to persist for years. The environmental conditions that allowed prototaxites to grow so large were unique to that time. The atmosphere was different: lower oxygen levels, higher carbon dioxide, a climate that was generally warm and humid. The land was still being colonized, and there was little competition for space, for light, for resources. The absence of herbivores — there were no land animals large enough to feed on prototaxites — meant that the organisms could grow without the threat of being consumed. The lack of tall plants meant that prototaxites had access to unfiltered sunlight if it had photosynthetic partners, or to the full range of organic matter on the ground if it was saprotrophic. How did they grow so large? The mechanism is unclear, but it likely involved a combination of factors: abundant moisture, lack of competition, a slow but steady accumulation of biomass, and perhaps a unique structural adaptation that allowed the hyphae to interlock and support each other in ways that modern fungi cannot replicate. The growth was probably slow, measured in millimeters per year, but sustained over decades, the organism gradually increasing in height and girth, adding layer upon layer of hyphae, building a structure that was part organism, part architecture. The reproduction of prototaxites is also a mystery. No spores have been definitively identified in association with the fossils, though this may be due to the poor preservation of such delicate structures. It is assumed that prototaxites reproduced by releasing spores, either from the surface of the trunk or from specialized structures at the top, but the details are lost to time. The spores would have been carried by the wind, settling on moist ground, germinating, sending out hyphae that would eventually grow into new trunks, continuing the cycle. The extinction of prototaxites is as mysterious as its biology. It did not disappear suddenly, but gradually declined over millions of years, becoming less common, less widespread, until it vanished entirely from the fossil record, around 350 million years ago. The rise of forests is the most likely explanation. The new plants created new conditions, new competitors, new challenges that prototaxites could not overcome. The shade cast by trees would have reduced the light available for any photosynthetic partners. The accumulation of leaf litter would have changed the chemistry of the soil. The evolution of new fungi better adapted to decomposing wood would have outcompeted prototaxites for resources. Or perhaps prototaxites did not go extinct but evolved, transforming into something smaller, less conspicuous, less recognizable in the fossil record. Fungi are notoriously difficult to fossilize, and the absence of evidence is not evidence of absence. It is possible that the descendants of prototaxites are still among us, hidden in the soil, in the wood, in the mycelial networks that thread through the forest floor, their ancestry obscured by hundreds of millions of years of evolution. The story of Prototaxites is a story of a world that no longer exists, a time when fungi were giants, when the land was young, when the rules of ecology were still being written. It is a reminder that the present is not the only possibility, that life has taken forms we can barely imagine, that the history of the Earth is filled with experiments, with organisms that rose and fell, that dominated and disappeared, leaving behind only traces, only fossils, only questions. And in those fossils, in those ancient rocks, in the cross-sections of trunks that once stood 8 meters tall, we see a glimpse of a world where fungi ruled, where they were the tallest, the largest, the most visible organisms on land. We see a world before trees, before forests, before the green canopy that now covers so much of the earth. We see a world where the mycelium did not hide in the soil but rose into the air, building structures that defied the scale of everything around them, monuments to the power of fungi to shape the land, to colonize new environments, to adapt and persist. The giants are gone, but the fungi remain, smaller now, more diverse, more hidden, but no less essential, no less ancient, no less remarkable. They are the descendants of the pioneers, the inheritors of a billion-year legacy, the living representatives of a kingdom that has survived every mass extinction, every climate change, every upheaval that the earth has endured. And beneath the forest floor, where the light of the sun never reaches, the mycelium continues its work, growing, branching, connecting, recycling, supporting, a vast and silent intelligence that remembers in its chemistry, in its genes, in its structure, the time when fungi were giants, when they stood tall, when they ruled the land. The memory is encoded in the DNA, in the patterns of growth, in the strategies of survival that have been passed down through countless generations, through hundreds of millions of years, from Prototaxites to the mushrooms that fruit in the forest today. The giants are gone, but their legacy endures, written in the living tissue of every fungus that grows, every mycelium that spreads, every spore that drifts on the wind. And the earth remembers in its rocks, in its fossils, in the layers of sediment that preserve the shapes of organisms long extinct, the time when fungi were the tallest things on land, when they were the giants, when they ruled. Fungi do not only decompose and connect, they heal, they calm, they restore balance to systems that have fallen into disorder. For thousands of years, humans have turned to mushrooms for medicine, for compounds that reduce inflammation, that modulate the immune system, that protect the brain, that ease anxiety, that promote sleep. These are not superstitions or folk remedies, but biochemical realities, molecules shaped by millions of years of evolution, originally developed by fungi to defend themselves, to communicate, to survive, now repurposed by human bodies, fitting into receptors, altering pathways, influencing the chemistry of consciousness itself. The mushrooms do not heal with intention. They simply produce compounds, secondary metabolites, chemicals that serve the fungus in its struggle for survival. But these compounds, when consumed by humans, interact with our biology in ways that can be profound, therapeutic, transformative. Reishi, Ganoderma lucidum, the mushroom of immortality, has been used in traditional Chinese medicine for over 2,000 years, revered as a tonic for longevity, for vitality, for spiritual well-being. It grows on decaying hardwood trees, particularly oaks, its fruiting body a glossy, kidney-shaped bracket with a varnished surface that ranges from deep red to almost black, its texture woody, its flesh too tough to eat fresh but rich in bioactive compounds when dried and extracted. The mushroom is called lingji in Chinese, a name that translates to spirit plant or herb of spiritual potency, reflecting its status not merely as medicine but as a symbol of health, of peace, of connection to the natural world. The chemistry of reishi is complex, containing over 400 bioactive compounds, including polysaccharides, triterpenes, peptides, and sterols. The polysaccharides, particularly beta-glucans, are immunomodulators, molecules that can enhance or suppress immune responses depending on the context, helping the body maintain balance, preventing overreaction, reducing chronic inflammation. The triterpenes, especially ganideric acids, have anti-inflammatory and hepatoprotective properties, protecting the liver, reducing oxidative stress, modulating the activity of enzymes involved in detoxification. Reishi does not stimulate. It calms. It is an adaptogen, a substance that helps the body adapt to stress, that restores homeostasis, that brings systems back into equilibrium. Studies have shown that reishi can reduce anxiety, improve sleep quality, lower blood pressure, and enhance overall well-being, though the mechanisms are not fully understood. The effects are subtle, cumulative, not the immediate relief of a pharmaceutical drug, but the gradual restoration of balance, the slow unwinding of tension, the quiet support of the body's own healing processes. The traditional preparation of reishi involves slicing the dried mushroom into thin pieces and simmering them in water for hours, extracting the water-soluble polysaccharides and some of the triterpenes, creating a dark, bitter tea that is consumed daily, sometimes for months, as a tonic. The taste is intensely bitter, earthy, almost medicinal, a flavor that speaks of the forest, of decay, of the deep chemistry of wood and fungus. Some traditions add honey or other herbs to balance the bitterness, but the bitterness itself is considered part of the medicine, a reminder that healing is not always pleasant, that transformation requires accepting what is difficult. The mushroom grows slowly, taking months to mature, its mycelium spreading through the wood, secreting enzymes, absorbing nutrients, accumulating the compounds that will eventually be concentrated in the fruiting body. The fruiting body itself can persist for years, growing larger each season, its surface becoming more glossy, its color deepening, its chemistry evolving. Harvesting reishi is a patient practice, waiting for the right moment when the mushroom has reached maturity, but has not yet begun to decay. When the concentration of bioactive compounds is at its peak, lion's mane Hericium erinaceus is different, a cascade of white, icicle-like spines that hang from dead or dying hardwood trees, resembling a lion's mane or a frozen waterfall. Its appearance so distinctive that it cannot be mistaken for any other mushroom. The fruiting body is soft, almost spongy, with a delicate flavor when cooked, but its true value lies not in its culinary appeal, but in its neurological effects, its ability to stimulate the production of nerve growth factor, a protein essential for the growth, maintenance, and survival of neurons. Nerve growth factor, NGF, is critical for brain health. It supports the survival of existing neurons, promotes the growth of new ones, and facilitates the formation of synapses, the connections between neurons that underlie learning, memory, and cognition. As we age, the production of NGF declines, and this decline is associated with cognitive impairment, memory loss, and neurodegenerative diseases like Alzheimer's and Parkinson's. Lion's mane contains compounds, hericenoids and erinacines, that can cross the blood-brain barrier and stimulate the production of NGF, potentially protecting the brain, enhancing cognitive function, and slowing the progression of neurodegeneration. The research is still emerging, but the results are promising. Studies in animals have shown that lion's mane can improve memory, reduce anxiety and depression, and promote the regeneration of damaged nerves. Human studies, though limited, have found improvements in cognitive function in older adults with mild cognitive impairment, reductions in anxiety and irritability, and enhanced overall well-being. The effects are not dramatic, not the sudden clarity of a stimulant, but a gentle enhancement, a subtle sharpening of focus, a quieting of mental noise. Lion's mane grows in temperate forests, fruiting in late summer and autumn, its white spines emerging from wounds in the bark of oak, beech, maple, and other hardwoods. The mycelium colonizes the heartwood, breaking down lignin and cellulose, recycling the tree's nutrients, and in the process, producing the compounds that support its own survival and incidentally the health of human brains. The mushroom is both decomposer and healer, breaking down the old, supporting the new, participating in cycles of decay and renewal that extend beyond the forest into the realm of human health. The medicinal properties of mushrooms are not accidents but adaptations, evolutionary solutions to the challenges fungi face in their environments. The compounds that reduce inflammation in human bodies also protect fungi from bacterial infections, from predators, from competitors. The polysaccharides that modulate our immune systems help fungi resist attack by other organisms. The antioxidants that protect our cells from oxidative stress also protect fungal cells from the reactive oxygen species generated during decomposition, during metabolism, during the chemical warfare that occurs in the soil. Fungi live in hostile environments, surrounded by bacteria, by other fungi, by insects and nematodes that would consume them if given the chance. They defend themselves with chemistry, producing antibiotics, toxins, repellents, signaling molecules. Penicillin, one of the most important antibiotics in human medicine, was originally a fungal defense compound produced by Penicillium species to suppress bacterial competitors. The same is true for many other medicinal compounds. They evolved not to heal humans, but to help fungi survive, and we have learned to harness them, to extract them, to use them for our own purposes. The relationship between humans and medicinal mushrooms is ancient, predating written history. Archaeological evidence suggests that humans have been using fungi for medicine for thousands of years, perhaps tens of thousands. The famous Iceman, a mummified body discovered in the Alps and dated to over 5,000 years ago, was carrying two species of mushrooms, one likely used as tinder, the other Fomitopsis betulina, known for its anti-parasitic properties, possibly used to treat intestinal parasites. Traditional medicine systems around the world, Chinese, Japanese, Indian, European, have long recognized the medicinal value of mushrooms, using them to treat infections, to boost vitality, to calm the mind, to extend life. Modern science is now validating many of these traditional uses, identifying the active compounds, elucidating the mechanisms, conducting clinical trials. But the science is still catching up to the wisdom of traditional practice, still discovering what herbalists and healers have known for centuries, that mushrooms contain molecules of remarkable complexity and potency, that they can influence human health in profound ways, that they are allies in the ongoing struggle to maintain balance, to resist disease, to live well. Turkey tail Trametes versicolor is another medicinal mushroom, a common bracket fungus that grows on dead wood, its fruiting body a thin leathery fan with concentric zones of colour, brown, grey, blue, green, orange, resembling the tail feathers of a wild turkey. It is one of the most extensively studied medicinal mushrooms, particularly for its immune-modulating and anti-cancer properties. The polysaccharide K, PSK, and polysaccharide peptide, PSP, extracted from turkey tail, have been used as adjunct cancer therapies in Japan and China for decades, shown to enhance the effectiveness of chemotherapy, reduce side effects, and improve survival rates in some cancers. Turkey tail does not kill cancer cells directly. It modulates the immune system, enhancing the activity of natural killer cells, macrophages, and T cells, helping the body recognize and destroy abnormal cells. It also has antioxidant and anti-inflammatory properties, reducing the chronic inflammation that can promote cancer growth. The mushroom is abundant, easy to find, growing on logs and stumps in forests around the world. Its colourful fans overlapping in dense clusters, each one releasing millions of spores, continuing the cycle. Chaga, Inonotus obliquus, grows on birch trees in cold climates. Siberia, Northern Europe, Canada, Alaska, forming black, crusty masses that protrude from the bark like burnt charcoal. The exterior is hard, almost rock-like, but the interior is softer, rust-coloured, rich in melanin, polysaccharides and triterpenes. Chaga has been used in Russian and Scandinavian folk medicine for centuries, brewed as a tea to treat stomach ailments, to boost immunity, to increase vitality. Modern research has found that chaga has potent antioxidant properties, among the highest of any natural substance, and that it can modulate the immune system, reduce inflammation, and inhibit the growth of cancer cells in laboratory studies. The mushroom grows slowly, taking years to develop, drawing nutrients from the living birch tree, gradually forming the conch that will eventually be harvested. The relationship is parasitic. The fungus weakens the tree, eventually killing it. But the tree can survive for years, even decades, with chaga growing on it, the two organisms locked in a slow struggle, the fungus extracting resources, the tree resisting, both persisting until the balance tips and the tree falls, providing the fungus with a new substrate to decompose. The use of medicinal mushrooms is not without caution. Some mushrooms are toxic, some are allergenic, and even medicinal mushrooms can have side effects or interact with medications. Reishi can thin the blood, potentially interacting with anticoagulants. Lion's mane can cause digestive upset in some people. Chaga, if consumed in large quantities, can cause kidney damage due to its high oxalate content. The key is moderation, knowledge, respect for the power of these organisms, recognition that they are not inert substances but living chemistry, evolved for purposes other than human health, yet capable of influencing it in profound ways. There are other medicinal mushrooms, each with its own chemistry, its own history, its own applications. Cordyceps, a parasitic fungus that infects insects, has been used in Tibetan and Chinese medicine for centuries to enhance energy, endurance, and respiratory function. The fungus colonizes the insect's body, consuming it from within, eventually erupting from the host as a slender, club-shaped fruiting body. Wild cordyceps, harvested from the high plateaus of the Himalayas, is among the most expensive natural substances in the world, but cultivated varieties are now available, offering similar benefits at a fraction of the cost. Studies suggest that cordyceps can improve athletic performance, increase oxygen utilization, and support kidney and liver function. Maitake grifola frondosa, known as hen of the woods for its ruffled, layered appearance, grows at the base of oak trees and has been shown to have immune-modulating and blood sugar-regulating properties. Shiitake lentinula edodes, one of the most widely cultivated mushrooms in the world, contains lentinin, a polysaccharide with anti-cancer and antiviral effects. The list goes on, each mushroom a different chemistry, a different set of compounds, a different potential for healing. The chemistry of peace that mushrooms offer is not the peace of sedation, not the numbing of symptoms, but the peace of balance, of systems functioning as they should, of inflammation reduced, of neurons protected, of immune responses modulated. It is the peace that comes from alignment with natural processes, from consuming substances that have co-evolved with the ecosystems we are part of, from recognizing that health is not the absence of disease but the presence of resilience, adaptability, harmony. And in the quiet of the forest, where reishi grows on oak, where lion's mane cascades from maple, where turkey tail fans across fallen logs, where chaga clings to birch, the fungi continue their work, producing compounds, defending themselves, decomposing wood, recycling nutrients, participating in the ancient cycles of life and death, growth and decay, offering to those who know how to listen, how to harvest, how to prepare, a chemistry of calm, a molecular peace, a reminder that healing is not separate from nature but embedded in it, waiting in the mycelium in the fruiting body, in the slow, patient work of organisms that have been here far longer than we have, that will be here long after we are gone. The mushrooms do not promise immortality, despite the name given to reishi. They promise only what they have always offered, compounds shaped by evolution, molecules that interact with biology, chemistry that can support, that can calm, that can restore, if used wisely, if approached with respect, if understood not as magic but as the accumulated wisdom of 1 billion years of fungal survival, distilled into the flesh of a mushroom, waiting to be discovered, to be consumed, to be integrated into the ongoing story of human health. In the deep darkness of the forest, where the canopy blocks the stars and the moon is only a rumour, there is sometimes a light, faint, ghostly green, emanating from decaying wood, from fallen logs, from the forest floor itself, a glow so subtle that it can only be seen when the eyes have fully adjusted to the dark, when all other sources of light have been extinguished. This is foxfire, the cold light of bioluminescent fungi, a phenomenon that has mystified humans for millennia, inspiring folklore, superstition, and scientific inquiry, a light produced not by heat, not by combustion, not by any external source, but by a chemical reaction within the cells of the fungus, a reaction that converts chemical energy directly into light with almost no waste heat, an efficiency that far exceeds any technology humans have yet devised. The light is alive. It is the glow of metabolism, of enzymes working, of molecules transforming, of energy flowing through living tissue. Bioluminescence in fungi is rare. Only about 80 species out of over 100,000 known fungi produce light, but those that do are found across the world, in tropical rainforests, in temperate woodlands, in subtropical regions, growing on dead wood, on leaf litter, on living trees. The most famous is Armillaria melea, the honey mushroom, whose mycelium glows faintly in the dark, creating an eerie luminescence in the wood it colonizes. Another is Omphalotus olearius, the jack-o'-lantern mushroom, which grows in clusters at the base of trees and stumps, its orange caps glowing green in the darkness, bright enough to read by if the eyes are fully dark adapted. The mechanism of bioluminescence in fungi involves a molecule called luciferin and an enzyme called luciferase. When luciferin is oxidized by luciferase in the presence of oxygen, it produces light, photons emitted as the molecule returns from an excited state to its ground state. The reaction is highly efficient, with nearly 100% of the energy converted into light rather than heat, a cold light, a living light, a light that does not burn. But why do fungi glow? The question has puzzled scientists for decades, and the answer is still not entirely clear. Unlike bioluminescence in other organisms, fireflies use it to attract mates, deep-sea fish use it to lure prey or communicate in the darkness. The function of fungal bioluminescence is less obvious. The fungi do not have eyes, do not respond to their own light, do not seem to use it for communication in any direct sense. One hypothesis is that the light attracts insects, which then disperse the spores. Studies have shown that bioluminescent mushrooms do attract more insects than non-luminescent mushrooms, and that the insects that visit them can carry spores on their bodies, spreading the fungus to new locations. The light would be a signal, a beacon in the darkness, drawing in the dispersal agents, ensuring that the spores are carried far from the parent organism, increasing the chances of colonization, of reproduction, of survival. Another hypothesis is that bioluminescence is a byproduct of metabolism, a side effect of the oxidative reactions that occur during decomposition, during the breakdown of lignin and cellulose, during the detoxification of reactive oxygen species. The light would be incidental, not adaptive, a consequence of the chemistry of decay rather than a strategy for survival. But this seems unlikely given the energy cost of producing luciferin and luciferase, the specificity of the reaction, the fact that bioluminescence is found in multiple, unrelated lineages of fungi, suggesting that it has evolved independently multiple times, which would be surprising if it served no function. A third hypothesis, more speculative, is that the light serves a defensive function, deterring predators or competitors. Some bioluminescent organisms produce light when disturbed, startling or confusing potential threats. It is possible that the glow of fungal mycelium serves a similar purpose, warning away insects, nematodes, or other organisms that might otherwise consume the fungus. But the evidence for this is limited, and the hypothesis remains unproven. The truth may be a combination of these factors, or something else entirely. The mystery of foxfire persists, a reminder that even the most studied organisms still hold secrets, that the natural world is full of phenomena we do not yet fully understand, that the glow in the darkness is not just light, but a question, waiting for an answer. The chemistry of the reaction is elegant in its simplicity, yet complex in its execution. The luciferin molecule in fungi is different from the luciferin in fireflies or in marine organisms. Each lineage has evolved its own version, its own pathway, its own chemistry of light. In fungi, the luciferin is thought to be a derivative of caffeic acid, a compound found in many plants and fungi, modified and oxidized by the luciferase enzyme to produce the characteristic green glow. The reaction requires oxygen, which is why bioluminescence is often strongest near the surface of the mycelium, where oxygen is most available, and why it can be enhanced by gently blowing on glowing wood, increasing the oxygen supply, brightening the glow momentarily before it fades back to its baseline level. The glow is not uniform across the mycelium, but concentrated in certain areas. The growing tips of hyphae, the regions of active metabolism, the zones where decomposition is most intense. This creates patterns, constellations of light within the wood, networks of luminescence that mirror the branching structure of the mycelium itself. Under a microscope, individual hyphae can be seen glowing tiny threads of light woven through the substrate, each one a filament of living fire, cold and green and patient. The experience of seeing bioluminescent fungi in the wild is unforgettable. It requires patience, darkness, and luck. The glow is faint, easily overwhelmed by even the smallest amount of ambient light, a flashlight, a phone screen, the distant glow of a city. To see it, one must venture into the forest at night, allow the eyes to adjust for at least 20 minutes, and search for the telltale green glow on fallen logs, on stumps, on the forest floor. The light is not constant but flickers, pulses, varies in intensity depending on the age of the mycelium, the moisture, the temperature, the metabolic activity of the fungus. Some who have witnessed foxfire describe it as otherworldly, as if the forest itself is breathing light, as if the boundary between the living and the dead has become permeable, as if the wood remembers being a tree and glows with the memory. Others describe it more prosaically, as a curiosity, a natural phenomenon, interesting but not transcendent. But even the most skeptical observer cannot deny the strangeness of it, the uncanniness of light emerging from decay, of photons born from rot, of beauty arising from decomposition. The history of foxfire is woven into human culture, appearing in folklore, in literature, in the accounts of travelers and naturalists who encountered the phenomenon and struggled to explain it. In medieval Europe, glowing wood was sometimes called fairy fire or will-o'-the-wisp, attributed to supernatural forces, to spirits, to the souls of the dead. Sailors reported seeing glowing wood on ships, a phenomenon that was both eerie and practical. The light was sometimes used for illumination in the dark holds of ships, a natural lantern that required no fuel, no flame, no maintenance. Aristotle wrote about luminous wood, noting that it glowed without heat, a mystery that defied the understanding of his time. Pliny the Elder described glowing fungi in his Natural History, speculating about their nature, but offering no explanation. It was not until the 19th century, with the development of microscopy and biochemistry, that scientists began to understand the mechanism, identifying the luciferin-luciferase reaction, isolating the compounds, studying the conditions under which the light was produced. The study of bioluminescence has practical applications beyond understanding fungi. The luciferin-luciferase system has been adapted for use in biotechnology, in medical research, and environmental monitoring. Luciferase is used as a reporter gene, a marker that allows scientists to track gene expression, to visualize cellular processes, to detect the presence of specific molecules. The enzyme is fused to a gene of interest, and when the gene is active, the enzyme produces light, which can be measured with sensitive detectors. This technique has been used to study cancer, to track infections, to monitor the effectiveness of drugs, to understand the fundamental processes of life at the molecular level. Bioluminescent fungi have also inspired artists, writers, and designers who see in the glow a symbol of the hidden life of the forest, of the beauty that exists in darkness, of the connection between decay and renewal. The light is a reminder that decomposition is not just destruction but transformation, that the breaking down of dead matter releases energy, that even in death there is a kind of vitality, a glow that persists until the last molecules are consumed, the last reactions complete, the last photons emitted. The diversity of bioluminescent fungi is remarkable. Mycen species, small mushrooms that grow on leaf litter and decaying wood, are among the most common bioluminescent fungi, their caps and stems glowing faintly in the dark. Panellus stypticus, found in North America and Europe, grows on dead hardwood and produces a greenish glow from its gills. In the tropics, species like Neonithoponus gardneri produce a brighter light, visible even from a distance, their large, fan-shaped caps glowing with an intensity that can illuminate the surrounding vegetation. The intensity of the glow varies with environmental conditions. Bioluminescence is strongest in warm, humid conditions, when the metabolic activity of the fungus is high, when the enzymes are working efficiently, when the substrates are abundant. In cold or dry conditions, the glow diminishes, sometimes disappearing entirely, only to return when conditions improve. The light is a reflection of the fungus's vitality, a visible manifestation of its metabolic state, a signal that the organism is alive, active, engaged in the work of decomposition. The glow also follows a circadian rhythm in some species, brightest at night, dimmer during the day, even though the fungus is in complete darkness and has no direct way of sensing sunlight. This suggests an internal clock, a biological rhythm that persists even in the absence of external cues, a memory of day and night encoded in the chemistry of the organism. The mechanism is not fully understood, but it may be linked to the activity patterns of the insects that the light is thought to attract, which are themselves most active at night, making nighttime the optimal time for the fungus to glow, to signal, to recruit dispersal agents. The colour of fungal bioluminescence is almost always green, with a peak emission around 520 to 530 nanometers, in the middle of the visible spectrum, the wavelength to which the human eye is most sensitive. This is not a coincidence. The green light is the most efficient for penetrating the darkness, for being seen by insects, for serving whatever function the light evolved to serve. Other colours, blue, yellow, red, are rare or absent in fungal bioluminescent organisms. suggesting that the chemistry of fungal light is constrained by the specific luciferin and luciferase involved, by the evolutionary history of the fungi, by the ecological context in which the light is used. In some tropical forests, bioluminescent fungi create landscapes of light, entire hillsides glowing faintly in the darkness, the accumulated glow of thousands of individual mushrooms, of mycelial networks spreading through the leaf litter, of decomposition happening on a massive scale. Travelers in the Amazon, in Southeast Asia, in the rainforests of Australia have reported seeing these glowing forests, describing them as magical, as dreamlike, as places where the boundary between the natural and the supernatural seems to dissolve. The glow of foxfire is not just a curiosity, not just a beautiful phenomenon, but a window into the biochemistry of fungi, into the strategies they use to survive, to reproduce, to interact with their environment. It is a reminder that light is not only the province of the sun, of fire, of electricity, but can be produced by living organisms, by chemical reactions, by the slow, patient work of enzymes transforming molecules in the darkness, and in the forest where the night is deep and the silence is profound, where the only sounds are the rustle of leaves and the distant call of an owl. The glow of bioluminescent fungi is a presence, a companion, a light that asks nothing, that offers only itself, a cold green glow that has been shining for millions of years, long before humans walked the earth, long before there were eyes to see it, a light that will continue to shine long after we are gone, as long as there are fungi, as long as there is wood to decompose, as long as there is darkness to illuminate. The mystery of foxfire is not fully solved, but the search for answers continues in laboratories, in forests, in the minds of scientists and naturalists who are drawn to the glow, who want to understand not just how it works, but why it exists, what purpose it serves, what story it tells about the evolution of fungi, about the ecology of decomposition, about the hidden life of the forest. And for now, the light glows on, faint and ghostly, a beacon in the darkness, a reminder that the world is full of wonders, that the forest is alive in ways we cannot always see, that even in decay there is beauty, that even in darkness there is light, waiting to be discovered, waiting to be understood, waiting to be appreciated for what it is, a living glow, a chemical miracle, a gift from the fungi to the night. The foxfire burns without heat, shines without flame, glows without end, as long as the mycelium lives, as long as the wood decays, as long as the forest stands. It is a light that belongs to the darkness, that exists because of the darkness, that will be invisible in the day, but in the night becomes a presence, a mystery, a wonder. And beneath the forest floor, where the light of the sun never reaches, the mycelium glows faintly, patiently, endlessly, a living constellation in the soil, a map of decay, a network of light that connects the dead wood to the living forest, the past to the present, the darkness to the glow. A single mushroom can produce billions of spores, tiny microscopic particles, each one a complete genetic package, a potential new organism, released into the air in numbers so vast that they defy comprehension, drifting on currents of wind, riding thermals, settling on surfaces miles from their origin, waiting for the right conditions to germinate, to send out hyphae to begin the cycle again. The spores are everywhere, in every breath of air, on every surface, invisible to the naked eye but present in staggering abundance, a constant rain of potential life falling silently through the atmosphere, most of them doomed to land in inhospitable places, to dry out, to be consumed, to fail, but a few, just a few, finding the right substrate, the right moisture, the right temperature, and beginning to grow. This is the wisdom of spores, not intelligence, not foresight, but a strategy refined by hundreds of millions of years of evolution, a strategy of abundance, of dispersal, of patience. The fungus does not know where its spores will land. It cannot direct them, cannot choose the destination. It can only produce them in vast numbers and release them, trusting to chance, to wind, to the laws of probability that somewhere, somehow some of them will find a place to grow. The production of spores is energetically expensive. A large mushroom, a giant puffball, a bracket fungus, a cluster of honey mushrooms can devote the majority of its resources to spore production, creating trillions of spores over its lifetime, each one requiring proteins, lipids, carbohydrates, pigments, cell walls. The investment is enormous, but the payoff when it comes can be equally vast. A single successful spore can give rise to a mycelial network that spans acres, that persists for centuries, that produces its own fruiting bodies and releases its own spores, continuing the lineage. The spores themselves are marvels of engineering, designed to survive harsh conditions, to remain viable for years, sometimes decades, waiting for the right moment to germinate. They are encased in thick walls, often pigmented to protect against ultraviolet radiation, resistant to desiccation, to heat, to cold, to chemical attack. Some spores can survive being frozen, being heated to near boiling temperatures, being exposed to intense sunlight, and still retain the ability to germinate when conditions improve. The size of spores varies widely among species, but most are between 3 and 20 micrometers in diameter, small enough to remain airborne for extended periods, light enough to be carried by the slightest breeze. The smaller the spore, the farther it can travel, the longer it can stay aloft, but also the more vulnerable it is to drying out, to being damaged by radiation, to being consumed by bacteria or other microorganisms. There is a trade-off between dispersal and survival, and different fungi have evolved different strategies, different sizes, different shapes, different defenses. The shape of spores is also highly variable: spherical, elliptical, cylindrical, star-shaped, ornamented with spines or ridges or warts, smooth or rough, each shape adapted to a particular mode of dispersal, a particular ecological niche. Some spores are sticky, designed to adhere to insects or animals that brush against the fruiting body, hitching a ride to new locations. Others are hydrophobic, repelling water, preventing them from clumping together, ensuring that they disperse individually rather than in masses. Still others are equipped with appendages, with tails or wings or hooks, that help them catch the wind, that orient them in the air, that increase their chances of landing in a favorable position. The colors of spores range from white to black, from yellow to purple, from pink to brown, each pigment serving a purpose: protection from ultraviolet radiation, deterrence of predators, chemical signaling, or simply a byproduct of the compounds used to construct the spore wall. The spore print, the deposit of spores left when a mushroom cap is placed on paper overnight, reveals the color and can be used to identify species, to distinguish between similar-looking mushrooms, to understand the diversity hidden in what appears to the eye as a single type of fungus. The release of spores is often synchronized with environmental conditions: humidity, temperature, air currents. Many mushrooms release spores at night when the air is calm and moist, when the spores are less likely to dry out, when the conditions are optimal for dispersal. Others release spores in response to rain, to changes in barometric pressure, to vibrations caused by animals or falling debris. The timing is critical, and fungi have evolved mechanisms to sense the environment, to wait for the right moment, to maximize the chances that their spores will find a suitable home. Some fungi have evolved relationships with specific dispersal agents. Truffles, which fruit underground, produce intensely aromatic compounds that attract animals: wild boars, deer, rodents, even insects, that dig up the fruiting bodies, consume them, and disperse the spores in their feces, depositing them in new locations along with a package of nutrients to support germination. The relationship is mutualistic. The animal gets food, the fungus gets dispersal, and it has shaped the evolution of both partners, leading to the development of complex scent profiles, of timing mechanisms that ensure the truffles ripen when the animals are most active, of spore structures that can survive passage through the digestive system. The dispersal mechanisms are diverse, creative, sometimes astonishing. Puffballs release spores in clouds when the fruiting body is compressed, either by rain, by wind, by an animal stepping on it, or simply by the collapse of the aging structure. The spores puff out in a visible cloud, a brown or grey smoke that drifts on the breeze, settling slowly to the ground. A single large puffball can contain trillions of spores, and when it ruptures, the release is dramatic, a sudden exhalation of potential life. Bracket fungi, the shelf-like structures that grow on trees, release spores from pores or gills on the underside of the fruiting body, allowing gravity to assist in the dispersal. The spores fall from the pores, are caught by air currents beneath the bracket, and are carried away from the tree, away from the parent organism, into the wider world. The orientation of the bracket is critical. It must be horizontal, or nearly so, to ensure that the spores fall freely and are not trapped against the tree trunk. If the tree tilts, if the bracket grows at an angle, the fungus can adjust, reorienting its growth, ensuring that the spore-producing surface remains horizontal, that the dispersal continues. Some fungi use water to disperse their spores. Cup fungi, morels, and other Ascomyces produce spores in sac-like structures called asci, which build up internal pressure and then explosively discharge the spores into the air, launching them several centimeters away from the fruiting body. The discharge is so rapid, so forceful, that it can be heard as a faint hissing or popping sound if one listens closely in a quiet forest. The spores are then carried by wind by insects, by rain splash, to new locations. Birds' nest fungi have evolved an even more elaborate mechanism. The fruiting body resembles a tiny bird's nest, complete with eggs, small lens-shaped structures called peridioles that contain the spores. When a raindrop strikes the nest, the force of the impact ejects the peridioles, which are attached to a sticky cord that unravels as they fly through the air, wrapping around nearby vegetation and anchoring the peridioles in place. The spores are then released gradually as the peridioles break down, ensuring that they are deposited in a location where moisture is available, where conditions are favorable for germination. Stinkhorns take a different approach, producing fruiting bodies that smell like rotting flesh, attracting flies and other carrion-feeding insects. The spores are embedded in a sticky, foul-smelling slime that covers the cap of the stinkhorn. The insects land on the cap, feed on the slime, and in the process coat their bodies with spores, which they then carry to other locations, depositing them on decaying organic matter, on dung, on carcasses, wherever the insects go next. The strategy is effective, if unpleasant to human senses, and it ensures that the spores are delivered to substrates rich in nutrients, ideal for fungal growth. The distances that spores can travel are remarkable. Studies have found fungal spores in the upper atmosphere at altitudes of several kilometers, carried by high-altitude winds, capable of traveling hundreds or even thousands of miles before settling back to Earth. Spores have been found in the Arctic, in deserts, in the middle of oceans, far from any obvious source, testament to the power of wind dispersal, to the ubiquity of fungi, to the relentless spread of life across the planet. But distance is not the only measure of success. Many spores land close to the parent organism within meters or even centimeters, and this too can be advantageous. If the parent has found a suitable substrate, if it has thrived in a particular location, then the immediate vicinity is likely to offer similar conditions, similar opportunities. Local dispersal ensures that the offspring remain in a proven environment, but they do not waste energy searching for resources that may not exist elsewhere. The germination of a spore is a critical moment, a transition from dormancy to active growth, from potential to actual. The spore must sense the environment, must detect the presence of moisture, of nutrients, of the right temperature, and must make the decision to germinate or to remain dormant. The decision is irreversible. Once germination begins, the spore commits its resources, extends a germ tube, begins to grow, and if the conditions are not suitable, if the moisture disappears, if the nutrients are insufficient, the young hypha will die and the spore's potential will be lost. Fungi have evolved sophisticated mechanisms for sensing the environment, for ensuring that germination occurs only when conditions are favorable. Some spores require specific chemical signals, compounds released by decaying wood, by plant roots, by other fungi, that indicate the presence of a suitable substrate. Others require a period of dormancy, a cold treatment or a passage through an animal's digestive system before they can germinate, ensuring that they do not sprout prematurely, that they wait for the right season, the right conditions. The longevity of spores is remarkable. Some can remain viable for decades, even centuries, lying dormant in the soil, in the wood, in the air, waiting for conditions to change, for an opportunity to arise. Spore banks in the soil are reservoirs of fungal diversity, containing species that may not fruit for years, that remain hidden until a disturbance, a fire, a flood, a windfall, creates the conditions they need. When a forest is cleared, when a field is ploughed, when a building is demolished, the spores that have been waiting in the soil can suddenly germinate, colonizing the disturbed ground, beginning the process of succession, of recovery, of renewal. This dormancy is a form of time travel, a way for fungi to persist through unfavorable periods, to outlast droughts, to survive winters, to wait for the return of the conditions that allow them to thrive. The spore is a seed of patience, a capsule of potential that can endure, that can wait, that can persist until the moment is right. The wisdom of spores is the wisdom of patience, of abundance, of accepting failure as part of the strategy. Most spores will not germinate. Most will land in places where they cannot survive. But the few that do succeed, that find the right substrate, that grow into mycelial networks, that produce their own fruiting bodies, will carry the lineage forward, will continue the cycle, will release their own billions of spores into the wind, perpetuating the strategy, ensuring that fungi remain ubiquitous, diverse, essential. And in the air, invisible, uncountable, the spores drift, a constant presence, a living dust, a reminder that life is not confined to the visible, to the tangible, but exists in forms so small, so numerous, so patient, that they escape our notice, yet shape the world in profound ways. The spores are the future, the potential, the seeds of forests yet to grow, of wood yet to decay, of cycles yet to turn. They fall like snow, like rain, like the slow, silent accumulation of time itself, each one a possibility, a chance, a gamble on the future. And somewhere, in the vastness of the world, a few will land in the right place at the right time, and will begin to grow, sending out hyphae, forming mycelium, connecting to the network, joining the ancient conversation that has been ongoing for 1 billion years, adding their voice to the chorus, continuing the work. The wisdom of spores is not a plan, not a design, but an acceptance of uncertainty, a strategy that embraces randomness, that turns the vastness of the world into an opportunity rather than an obstacle. The fungus does not control where its spores will go, but it ensures that they go everywhere, that they are present in every corner of the earth, waiting, patient, ready to grow when the moment comes. And the moment always comes, eventually, somewhere, for some of them. And that is enough. The relationship between trees and fungi is older than forests, older than flowers, older than the first insects took flight, a partnership that began over 400 million years ago, when the first plants ventured onto land and found the soil barren, devoid of nutrients, hostile to roots that had evolved in water in the gentle currents of ancient seas. The plants could not survive alone. They needed help, and the fungi, already established on land, already adept at extracting nutrients from rock and organic matter, offered a solution, a trade, an exchange, a symbiosis that would shape the evolution of terrestrial life, that would make forests possible, that would create the green world we know today. This is mycorrhiza, the fungus root, a fusion of plant and fungal tissue so intimate that the two organisms function as one, sharing resources, sharing signals, sharing survival. The fungus colonizes the roots of the plant, extending its hyphae into the soil, increasing the surface area for absorption by hundreds, sometimes thousands of times, reaching nutrients and water that the roots alone could never access. In exchange, the plant provides the fungus with sugars, the products of photosynthesis, compounds that the fungus cannot produce on its own because it lacks chlorophyll, because it cannot capture the energy of the sun. The exchange is not optional. Over 90% of plant species form mycorrhizal associations, and many cannot survive without them. The plants are dependent, reliant on the fungi to provide phosphorus, nitrogen, trace minerals, water, especially in poor soils, in dry climates, in environments where resources are scarce. The fungi, in turn, are dependent on the plants, receiving up to 30% of the carbon that the plant fixes through photosynthesis, a massive transfer of energy from the green world to the hidden world beneath the soil. There are two main types of mycorrhizae, ectomycorrhizae and arbuscular mycorrhizae, each with its own structure, its own ecology, its own evolutionary history. Ectomycorrhizae form a sheath around the root tips, a mantle of fungal tissue that encloses the root, with hyphae extending outward into the soil and inward between the root cells, creating a network called the Hartig net. This type of mycorrhiza is common in temperate and boreal forests, associated with trees like pines, oaks, birches, beeches, and many others. The fungi involved are often the familiar mushrooms of the forest floor, boletes, amanitas, russulas, chanterelles, species that fruit above ground, releasing spores, continuing the cycle. Arbuscular mycorrhizae are older, more widespread, found in association with the majority of plant species, including most crops, grasses, and herbaceous plants. The fungi penetrate the root cells, forming branching structures called arbuscules that resemble tiny trees, increasing the surface area for exchange, facilitating the transfer of nutrients from fungus to plant and carbon from plant to fungus. The fungi involved belong to a single ancient lineage, the Glomeromycota, and they do not produce large fruiting bodies remaining hidden in the soil, invisible except under a microscope, yet essential to the productivity of ecosystems, to the fertility of soils, to the growth of the plants that feed the world. The arbuscules are ephemeral, lasting only a few days before being digested by the plant cell, their nutrients absorbed, their structure broken down, but new arbuscules are constantly forming, the fungus constantly colonizing new cells, the exchange constantly renewed. The relationship is dynamic, responsive, adjusting to the needs of both partners, to the availability of resources, to the changing conditions of the soil. The specificity of mycorrhizal relationships varies. Some fungi are generalists, capable of forming associations with many different plant species. Others are specialists, forming relationships with only a few species, sometimes only one. The truffles, for example, are highly specific, associating with particular trees, oaks, hazels, pines, and they cannot complete their life cycle without their hosts. This specificity has implications for conservation, for forestry, for the cultivation of these valuable fungi, because the success of the fungus depends on the presence of the right host, and the success of the host often depends on the presence of the right fungus. The mycorrhizal network is not just a one-to-one relationship between a single plant and a single fungus. It is a web, a network, connecting multiple plants to multiple fungi, creating a system of exchange and communication that spans the forest, that links trees of different species, different ages, different sizes. A single fungal individual can connect to dozens of plants, and a single plant can be colonized by dozens of fungal species, creating a complex, dynamic network where resources flow in multiple directions, where the health of one organism affects the health of others, where the forest functions as a superorganism greater than the sum of its parts. The network allows for the transfer of carbon between trees. Older, larger trees with more extensive root systems and more access to sunlight can share carbon with younger, smaller trees growing in the shade, supporting them, helping them survive until they can reach the canopy, until they can photosynthesize on their own. This transfer is not altruism but a form of investment, a strategy that maintains the diversity and resilience of the forest, that ensures that the next generation of trees is ready to replace the old when they fall, when they die, when the canopy opens and light floods the forest floor. Research by Suzanne Simard and others has shown that this transfer is substantial, that mother trees, the largest, oldest trees in the forest, can support dozens of younger trees, that the network creates a kind of social structure, a hierarchy, a system of mutual support that increases the survival of seedlings, that accelerates the recovery of forests after disturbances, that makes the ecosystem more resilient to drought, to disease, to climate change. The network also allows for the transfer of information. When a tree is attacked by insects, it releases chemical signals into the air and into the mycorrhizal network, warning neighboring trees, which respond by activating their defenses, producing compounds that deter herbivores, that attract predators of the herbivores, that make the leaves less palatable, less nutritious. The signal travels faster through the network than through the air, reaching trees that are shaded, that are downwind, that would not otherwise receive the warning. The mycorrhizal network is a communication system, a living internet, a way for plants to share information, to coordinate responses, to survive in a world full of threats. The mycorrhizal relationship is ancient, but it is not static. It evolves, adapts, responds to changing conditions. In nutrient-rich soils, plants may reduce their investment in mycorrhizae, allocating less carbon to the fungi, relying more on their own roots. In nutrient-poor soils, the investment increases, the relationship deepens, the dependence becomes more pronounced. The balance shifts depending on the environment, on the availability of resources, on the needs of the partners. The relationship is also not always mutualistic. Under certain conditions, it can become parasitic, with one partner taking more than it gives. Some plants, called mycoheterotrophs, have lost the ability to photosynthesize and rely entirely on the mycorrhizal network for carbon, tapping into the network, stealing sugars from other plants, living as parasites on the system. Indian pipe, ghost plant, and other achlorophyllous plants are examples, pale, ghostly organisms that emerge from the forest floor, flower briefly, and then disappear, their entire existence dependent on the hidden network beneath the soil. Some fungi, too, can be parasitic, taking carbon from plants without providing adequate nutrients in return, exploiting the relationship, weakening the host. The line between mutualism and parasitism is not always clear, and the relationship can shift depending on the context, on the health of the partners, on the availability of resources. The mycorrhizal network is not a utopia, but a complex dynamic system where cooperation and competition coexist, Where the interests of the partners sometimes align and sometimes diverge, the importance of mycorrhizae extends beyond individual plants and fungi to entire ecosystems. Mycorrhizal networks influence soil structure, binding soil particles together, reducing erosion, improving water retention. They influence nutrient cycling, mobilizing phosphorus and nitrogen from organic matter, making them available to plants, supporting productivity. They influence plant diversity, facilitating the coexistence of multiple species, creating niches, supporting rare and specialist plants that could not survive without their fungal partners. Mycorrhizal networks also play a role in carbon sequestration, in the storage of carbon in soils. The fungi produce glomalin, a glycoprotein that binds soil particles together and is highly resistant to decomposition, persisting in the soil for decades, storing carbon that would otherwise be released into the atmosphere. The mycelial networks themselves represent a significant carbon pool, containing vast amounts of carbon in their biomass, in their cell walls, in the compounds they produce. Protecting and enhancing mycorrhizal networks is a strategy for mitigating climate change, for increasing the capacity of soils to store carbon, for building resilience in ecosystems. The disruption of mycorrhizal networks has profound consequences. Deforestation, agriculture, urbanization, pollution, all can damage or destroy the networks, severing the connections, reducing the diversity of fungi, weakening the plants, degrading the soil. When a forest is cleared, the mycorrhizal fungi lose their hosts, and many species disappear, unable to survive without the carbon provided by the trees. When the forest is replanted, the fungi may not return, and the young trees struggle, growing slowly, suffering from nutrient deficiencies, vulnerable to drought and disease. Agricultural practices, particularly intensive tillage and the use of chemical fertilizers and pesticides, can also damage mycorrhizal networks. Tillage disrupts the hyphal networks, breaking the connections, reducing the effectiveness of the symbiosis. Chemical fertilizers can reduce the plant's dependence on mycorrhizae, leading to a decline in fungal diversity and abundance. Pesticides can directly harm the fungi, killing them or inhibiting their growth. The result is degraded soil, reduced productivity, increased reliance on external inputs, a system that is less sustainable, less resilient, less aligned with the natural processes that have sustained agriculture for millennia. The restoration of mycorrhizal networks is a key component of ecological restoration, of reforestation, of efforts to rebuild damaged ecosystems. Inoculating seedlings with mycorrhizal fungi before planting can improve survival rates, accelerate growth, increase resilience, protecting existing forests, maintaining connectivity, preserving the diversity of fungi. These are strategies for sustaining the networks, for ensuring that the sacred bond between trees and fungi persists, that the partnership that made forests possible continues into the future. The mycorrhizal relationship is also being explored for agricultural applications. Most crops form arbuscular mycorrhizae, and enhancing these relationships can reduce the need for fertilizers, improve drought tolerance, increase yields. Inoculating fields with mycorrhizal fungi, reducing tillage to preserve the networks, planting cover crops to support the fungi. These are practices that can make agriculture more sustainable, more resilient, more aligned with the natural processes that have sustained plant life for hundreds of millions of years. The sacred bond is not a metaphor, but a biological reality, a partnership so fundamental, so essential, that life on land as we know it would not exist without it. The trees reach toward the sun, capturing light, converting it into sugars, into energy, into the fuel that powers the forest. The fungi reach into the soil, dissolving rock, absorbing nutrients, recycling the dead, connecting the living. Together, they create the conditions for life, for diversity, for the complex, beautiful, resilient ecosystems that cover the land. And beneath the forest floor, where the light of the sun never reaches, the mycelium and the roots intertwine, exchanging molecules, sharing resources, communicating in a language of chemistry, of electrical signals, of flows and gradients that we are only beginning to understand. The bond is invisible, hidden, silent, but it is the foundation of the forest, the substrate upon which the visible world rests. The trees do not stand alone. They are supported, nourished, connected by the fungi, by the mycelial networks that thread through the soil, that link root to root, tree to tree, forest to forest. The relationship is ancient, evolved over hundreds of millions of years, refined by countless generations, tested by ice ages and droughts and fires and floods. And it has endured, persisted, adapted because it works, because it is beneficial, because it is essential. The sacred bond is a reminder that life is not a collection of isolated individuals, but a network of relationships, of exchanges, of dependencies. The tree needs the fungus needs the tree, and together they create something greater than either could achieve alone: a forest, a living system, a community of organisms bound together by invisible threads, by chemical signals, by the flow of nutrients and energy, by the ancient partnership that made the green world possible. And in the quiet of the forest, where the roots and the mycelium meet, where the exchange happens, where the bond is renewed with every molecule that passes from one organism to the other, the partnership continues, patient, persistent, essential, a testament to the power of cooperation, of symbiosis, of the hidden connections that sustain life on Earth. The sacred bond endures, as it has for 400 million years, as it will for as long as there are trees, as long as there are fungi, as long as the Earth remains a living world. The future is not made of steel and concrete, not built from plastics and petroleum, not dependent on the extraction of finite resources from a depleted Earth. The future, if it is to be a future worth living in, will be grown, cultivated, nurtured from living materials, from organisms that build without waste, that create without pollution, that return to the soil when their purpose is complete. The future is mycelium, the living architecture of fungi, a material that can be shaped, molded, directed to grow into forms that serve human needs. Building materials, packaging, textiles, even food, all produced by organisms that have been refining their craft for 1 billion years, that know how to transform simple substrates into complex structures, that work in partnership with the earth rather than against it. Mycelium-based materials are already being developed, tested, commercialized. Mushroom packaging grown from agricultural waste and fungal mycelium can replace polystyrene foam, providing cushioning and protection for shipped goods, then composting in a matter of weeks when discarded, returning nutrients to the soil rather than persisting in landfills for centuries. Mycelium bricks grown in molds can be used for construction, for insulation, for soundproofing, offering properties comparable to conventional materials but with a fraction of the environmental impact, produced at room temperature, requiring no kilns, no furnaces, no massive inputs of energy. Mycelium leather, grown from fungal cultures, offers an alternative to animal hides and synthetic plastics, a material that is durable, flexible, biodegradable, produced without the ethical concerns of animal agriculture, without the pollution of tanning processes, without the reliance on fossil fuels. The material can be grown in sheets, in any thickness, in any texture, tailored to specific applications, from fashion to furniture, from footwear to upholstery. The process is simple, elegant, almost miraculous in its efficiency. Agricultural waste, straw, sawdust, corn stalks, coffee grounds, is inoculated with fungal spores or mycelium. The mycelium colonizes the substrate, digesting it, binding it together with a network of hyphae, creating a solid, cohesive material. The growth is rapid, taking days to weeks depending on the application, and it can be stopped at any point by drying or heating, killing the mycelium, stabilizing the structure. The result is a material that is lightweight, strong, insulating, fire-resistant, and completely biodegradable, a material that embodies the principles of circular economy, of regenerative design, of working with nature rather than against it. Companies like Ecovative Design have pioneered the commercial production of mycelium materials, growing packaging, building materials, and even furniture from fungal cultures. Their process involves filling molds with agricultural waste and mycelium, allowing the fungus to grow for a few days, then harvesting and drying the material, creating products that are custom-shaped, fully compostable, and carbon negative. The mycelium sequesters more carbon during growth than is emitted during production and disposal. Mycelium composites can be tuned for different properties by varying the substrate, the fungal species, the growing conditions. A denser substrate produces a harder material, suitable for structural applications. A looser substrate produces a softer material, suitable for insulation or cushioning. Different fungal species produce different textures, different strengths, different rates of growth. The material is programmable, designable, adaptable to specific needs. Fungi are also being explored for bioremediation on a larger scale, for cleaning up contaminated sites, for breaking down pollutants, for restoring damaged ecosystems. Mycomediation, the use of fungi to degrade or sequester contaminants, has been applied to oil spills, to heavy metal contamination, to pesticide residues, to industrial waste. The fungi produce enzymes that can break down complex organic molecules, that can bind heavy metals, that can transform toxic compounds into less harmful forms. The process is slow, patient, thorough, requiring time and the right conditions, but it offers a solution that is biological, sustainable, aligned with the processes that have been cleaning and recycling the earth for billions of years. Paul Stamets, a mycologist and advocate for the potential of fungi, has demonstrated the use of mycelium to filter pollutants from water, to break down petroleum products, to remediate soil contaminated with heavy metals. His work has shown that fungi are not just decomposers of natural materials, but can be directed, cultivated, deployed to address the environmental damage caused by human activity, to heal the wounds we have inflicted on the earth. In one experiment, mycelium mats were used to filter runoff from a parking lot, removing petroleum hydrocarbons, heavy metals, and bacteria, producing water clean enough to support aquatic life. Fungi are also being explored as a source of sustainable food. Mycoprotein, derived from fungal cultures, is already used in meat substitutes, offering a high-protein, low-fat alternative to animal products produced with a fraction of the land, water, and energy required for livestock. The cultivation of edible mushrooms, oyster mushrooms, shiitake, lion's mane, and many others can turn agricultural waste into nutritious food, providing protein, vitamins, minerals, and medicinal compounds, supporting food security, reducing waste, creating livelihoods. The potential extends to material science, to medicine, to energy production. Fungi produce enzymes that can break down cellulose, that can convert plant biomass into sugars that can be fermented into biofuels. They produce compounds that have antibacterial, antiviral, anti-cancer properties that are being studied for pharmaceutical applications. They produce pigments, dyes, adhesives, all from renewable resources, all biodegradable, all part of the vision of a future where human industry is integrated with natural processes, where waste is minimized, where materials flow in cycles rather than linear paths from extraction to disposal. The return to the earth is not a retreat, not a rejection of technology, not a romanticization of the past. It is a recognition that the strategies that have sustained life for billions of years, cooperation, symbiosis, recycling, adaptation, are more sophisticated, more resilient, more sustainable than the strategies of extraction, exploitation, and waste that have characterized much of human industry. It is a recognition that fungi, with their billion-year history, with their mastery of decomposition, of nutrient cycling, of building complex structures from simple materials, have much to teach us, much to offer, if we are willing to listen, to learn, to work in partnership rather than domination. The mycelium does not compete for space, it fills the spaces between, connecting, supporting, recycling. It does not hoard resources. It shares, distributes, ensures that nutrients flow to where they are needed. It does not produce waste. Everything is used, everything is transformed, everything is returned to the cycle. These are principles that human systems can learn from, can emulate, can integrate into design, into manufacturing, into agriculture, into the way we live on this planet. The vision of a myco-future is not utopian but practical, grounded in the biology of fungi, in the research that is already being done, in the products that are already being made. It is a future where buildings are grown rather than built, where materials decompose when their use is complete, where pollution is remediated by living organisms, where food is produced in harmony with natural cycles, where the boundary between the human and the natural is not a wall, but a membrane, permeable, interactive, mutually beneficial. Architects and designers are already exploring mycelium as a building material. The myco tree, a structure built entirely from mycelium bricks, was exhibited in Seoul, demonstrating that fungal materials can support significant loads, can be shaped into complex forms, can create spaces that are beautiful, functional, and sustainable. The Living Pavilion, another mycelium structure, was grown in the Netherlands, its walls composed of mycelium and agricultural waste, its form organic, flowing, a demonstration of what architecture could become when it is grown rather than constructed. These are not distant visions but present realities, proof of concept projects that show what is possible when human creativity is aligned with fungal biology, when design works with natural processes rather than against them. The structures are temporary, biodegradable, returning to the soil when their purpose is complete, leaving no waste, no pollution, no permanent scar on the landscape. This future requires a shift in perspective, a recognition that humans are not separate from nature but part of it, that our survival depends on the health of the ecosystems we inhabit, that the fungi, the bacteria, the plants, the animals are not resources to be exploited, but partners to be respected, collaborators in the ongoing project of life on Earth. The shift is cultural as much as technological. It requires valuing regeneration over extraction, cooperation over competition, cycles over linear processes. It requires patience, humility, a willingness to learn from organisms that have been solving problems for far longer than humans have existed. It requires recognizing that the fastest, the newest, the most technologically advanced solution is not always the best, that sometimes the wisdom lies in the old, the slow, the living. The fungi have been here for over 1 billion years. They have survived 5 mass extinctions, countless climate changes, the rise and fall of countless species. They have adapted, evolved, persisted, not through domination but through cooperation, through symbiosis, through the patient work of decomposition and renewal. They are the custodians of the carbon cycle, the architects of the soil, the partners of plants, the healers, the recyclers, the hidden majority. And now, as humanity faces the consequences of its own success, as the climate changes, as resources dwindle, as ecosystems collapse, the fungi offer a path forward, a way to build, to heal, to sustain, that is aligned with the processes that have sustained life for eons. The myco-future is not a distant dream, but an emerging reality, visible in the laboratories where mycelium bricks are grown, in the fields where mushrooms are cultivated, in the forests where mycorrhizal networks connect trees, in the minds of scientists, designers, farmers, and visionaries who see in the fungi not just organisms but solutions, not just decomposers but builders, not just the past but the future. The return to the earth is a return to the wisdom of the mycelium, to the patience of spores, to the cooperation of symbiosis, to the cycles of growth and decay that have sustained the planet for billions of years. It is a recognition that the future is not something to be imposed on the earth, but something to be grown from it, nurtured by it, sustained by it. And beneath the forest floor, where the light of the sun never reaches, the mycelium continues its work, growing, branching, connecting, recycling, supporting. A vast and silent intelligence that has been breathing for 1 billion years, that has witnessed the rise and fall of empires, of species, of continents, that has persisted through ice ages and volcanic eruptions and asteroid impacts, that will persist long after we are gone, unless we choose to learn from it, to work with it, to become part of the network, part of the cycle, part of the living earth. The myco-future is not a rejection of humanity, but an invitation, an opportunity to align our ingenuity with the wisdom of the oldest kingdom, to build a civilization that does not extract and discard, but grows and recycles, that does not dominate but cooperates, that does not separate from nature but integrates with it. The fungi are waiting, patient as always, ready to share their secrets, ready to offer their services, ready to help us build a future that is sustainable, resilient, beautiful, if we are willing to listen, to learn, to grow. And in the quiet of the forest, where the mushrooms fruit and the mycelium spreads, where the spores drift on the wind and the networks connect tree to tree, the ancient partnership continues, the sacred bond endures, the cycle turns, and the earth, with its fungi, with its forests, with its hidden intelligence, breathes, lives, persists, offering to humanity, as it has offered to every organism that has ever lived, a place in the network, a role in the cycle, a chance to participate in the ongoing story of life. The myco-future is here, growing in the spaces between, waiting for us to notice, to understand, to join. And when we do, when we finally fall silent and listen, we will hear what the mycelium has been saying all along, that life is connection, that survival is cooperation, that the future is not built from steel and stone, but grown from the earth, nurtured by the oldest kingdom, sustained by the wisdom of 1 billion years. The return to the earth is the return to the mycelium, to the network, to the cycle, to the living intelligence that has been here all along, waiting for us to come home.